Hello everyone, I'm Anas Al Ubaidi, and this is lecture 23 or session 23 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of a drug validation and drug verification interaction trials. Introduction. Definitions and introduction. Definition for the drug validation interaction trials focus on evaluating whether a drug achieves its intended therapeutic effect in line with established goals, ensuring scientific validity. Drug verification interaction trials aim to confirm that the drug consistently produces the same outcomes under specified conditions, ensuring reliability. Key points. Patient safety, participant safety. Importance here, patient safety is the foremost priority during validation and verification phases. Effective monitoring ensures risks are promptly identified and managed, safeguarding trial participants while providing reliable evidence of the drug's safety profile. And here is a summarized diagram on ensuring patient safety in drug trials, left to right, starting from risk profiling, consistency monitoring, adverse event management, and then dosage adjustment. Key points or key actions for patient safety. Risk profiling in validation trials. Monitor common and severe adverse events, AEs, to compare observed risks with the expected safety profile. Consistency monitoring in verification trials detect discrepancies or protocol deviations that may harm participants. Example, a novel anticoagulant under validation triggers excessive bleeding in a subset of patients. Monitoring identifies the issue, enabling a dosage adjustment plan to mitigate the risk during verification. Challenges here: underreporting of adverse events, AEs in early phases, detection of rare side effects not evident during small sample validation trials. The outcome here, a focus on safety management, ensures the drug's risk-benefit ratio is favorable, enable, enabling progression to approval without ethical or regulatory barriers. Data integrity. The definition here as data integrity entails the accuracy, reproducibility, and credibility of all trial data across both validation and verification phases. Here is a summarized diagram on ensuring data integrity in clinical trials, starting from left to right: validated tools, cross-site reproducibility, source data verification challenges, and then outcomes. Still in data integrity, the key actions here: validated tools and systems, employ biomarker assays, validated devices, and electronic data capture EDC platforms to ensure accuracy. Example, data on a cholesterol-lowering drug is cross-validated during trials to confirm its initial effects, validation, and its consistent efficacy across multiple demographics, which is verification. Cross-site reproducibility: maintain consistent data collection protocols across trial sites for multi-site verification. Source data verifications, SDV, perform real-time audits to compare trial data with source documents for accuracy and completeness. So here as DV again, it is the verification of data, of the source, uh, the verification of the data of the source, and the way or this process uh, is simply the monitor or the auditor compare the data that is documented in the source for the site that is conducting the trial, and the data, compare this data in the source with the data that has been reported by that particular site to the electronic data capture to the EDC or to the sponsor or to the CRO. And here, the verification is to verify that this data is comparable and matchable between the source, what's in the source, and what has been reported. So source data verification. The challenge is here data fragmentation and inconsistencies due to differences in trial site capabilities and personnel. Variations in data interpretation by cross-location teams. The outcome here, robust data ensures the scientific validity and reproducibility of trial results, minimizing disputes and strengthening regulator confidence. Protocol adherence. Definition: strict adherence to trial designs ensures the elimination of variability and bias in drug outcomes, which is critical for validation and verification processes. The key actions here for protocol adherence: centralized monitoring systems use integrated dashboards to flag deviations in real time, training programs educate clinical staff on the trial's requirements to avoid protocol violations or protocol deviations, such as improper dosing regimens or irregular reporting schedules. Example, a diabetes drug trial disqualifies participants who fail to adhere to fasting protocols during validation, ensuring reliable glucose monitoring results. And the challenges here are difficulty maintaining compliance in decentralized or global trials, protocol amendments caused by emerging risk-benefit findings. The outcome here, adhering to defined protocols ensures the reliability, reproducibility, and regulatory acceptance of trial outcomes. And here is a summarized diagram on ensuring protocol adherence in clinical trials. From the left to the right, centralized monitoring, staff training, participant compliance, and overcoming challenges. Regulatory compliance, importance. Regulatory compliance ensures that all phases of validation and verification meet the ethical, scientific, and legal standards required by authorities such as the FDA, EMA, or ICH. FDA as of Food and Drug Administration Agency, EMA as of for European Medicines Agency, or ICH, which is the International Council for Harmonization. Key actions for regulatory compliance: targeted regulatory submissions. Submit findings showing the drug's intended effects during validation and reproducibility during verification. Proactive updates align with emerging regulatory requirements by regularly updating protocols and data packages. Example, a COVID-19 vaccine trial reports inconsistencies in immune response during verification, leading to an amendment of dosage protocols to align with emerging global safety standards. The challenge is here navigating differing regulations across countries for global trials, managing vast documentation requirements for regulator review. Outcome here: compliance ensures swift regulatory approvals, reducing delays and fostering global trust in the drug. Examples of successful monitoring: antihypertensive validation trials. The outcome here: confirmation of intended diastolic pressure reduction without adverse cardiovascular events. Osteoporosis verification trials. Outcome here: demonstrated consistent improvements in both bone density and fracture resistance across diverse age groups. COVID-19 vaccine trials. The outcome here: validation trials confirmed strong immune responses, while verification demonstrated consistent efficacy across geographies and demographics. And here's a diagram summarizing the overview of medical trials: COVID-19 vaccine trials, antihypertensive trials, and then osteoporosis trials. Summary tables, upcoming tables to summarize what we have covered through this lecture. And here's a table on key differences between drug validation and drug verification interaction trials. As here, in each aspect, there's comparison between both. As of definition, for drug validation interaction trial, evaluates if the drug achieves intended therapeutic effects, while drug verification interaction trial confirms the drug produces consistent outcomes under prescribed or specified conditions. And all the other aspects to compare, like main focus, objective, challenges, and example application. Table on key components of monitoring drug validation and verification trials. Component focus example, as in components, we have patient safety, data integrity, protocol adherence, regulatory compliance, endpoint validation. Table on tools and techniques for monitoring drug validation verification trials. Tool, technique, purpose example. Tools or techniques, we have EDC systems, centralized and remote monitoring, statistical analysis models, biomarker assessment tools, risk-based monitoring, RBM. Table on steps to effective monitoring for validation and verification. Step focus examples. Steps, we have site selection, RBM, risk-based monitoring, data review, reporting, adaptation. Table on real-world examples of successful monitoring applications, trial, scenario, purpose, outcome, trials or scenarios. We have antihypertensive trials, osteoporosis medication, chemotherapy drug, COVID-19 vaccine trials, and painkiller adolescent study. Application table, scenario, for each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios, we have monitoring safety parameters during an anticoagulant validation trial, cross-checking trial data for a cholesterol-lowering drug across multiple verification sites, enforcing strict participant fasting protocols in a diabetes drug verification trial, reporting immune response discrepancies in a verification trial for a vaccine, selecting spirometry-enabled clinical sites for asthma drug validation studies, monitoring adolescent trial participants for withdrawal effects in a painkiller verification trial, identifying gender-specific efficacy differences during a bone density drug validation trial, flagging blood pressure inconsistencies across verification centers for an anti-hypertensive drug, validating liver enzyme biomarkers for a hepatitis treatment, running regression analysis to confirm efficacy consistency in an osteoporosis drug verification trial. Next, we will revise what we have covered through this lecture through some multiple choice questions or MCQs. Question 1. What is the primary purpose of drug validation interaction trials? Um, A, to confirm the consistent application of research protocols. B, to evaluate whether a drug achieves its intended therapeutic effect. C, to determine manufacturing scalability. Or D, to bypass regulatory requirements. So definitely the answer is B, as to evaluate whether a drug achieves its intended therapeutic effect, as drug validation interaction trials are conducted to ensure that a drug meets its intended therapeutic goals, proving its efficacy and safety in specified uh, conditions. Question 2, why are drug verification interaction trials significant? Now, not the validation, the question about the verification. A, they eliminate the need for validation trials. B, they ensure consistent drug outcomes under specified conditions. C, they prioritize marketing strategies over trial safety. Or D, they focus solely on patient recruitment efforts. So the answer is B, as they ensure consistent drug outcomes under specified conditions. As the drug verification interaction trials confirm the reliability of outcomes by demonstrating consistency across conditions, which is critical for patient safety and regulatory approval. Question 3, how does risk-based management and monitoring, RBM, improve the trial process during drug verification phases? And actually, its purpose, the RBM, applies to any trial. A, it removes the need for patient safety assessment. B, it allocates resources toward high-risk populations and areas showing variability. C, it avoids the additional costs associated with regulatory filings. Or D, it focuses all resources equally across trial phases. So the answer is B, which applies to all trials, allocates. It allocates resources toward high-risk populations, prioritizes and areas showing variability, as RBM prioritizes monitoring efforts to high-risk aspects such as subgroups that show unexpected variability or adverse effects, ensuring efficient resource use while maintaining trial reliability. Question 4. Which tool is most effective for tracking reproducibility in drug validation verification trials? Is it A, for both interaction trials? A, statistical analysis model, B, manual documentation system, C, feedback surveys from participants, or D, randomized paper-based logs? So definitely it's A, statistical analysis models. As statistical analysis models help compare validation and verification results, ensuring consistent and reproducible trial outcomes across demographics and conditions. Question 5. What is the role of regulatory compliance in drug verification trials? A, to confirm that the trial meets ethical and legal standards for outcome consistency. B, to reduce protocol adherence requirements. C, to ensure patient recruitment strategies are streamlined. Or D, to eliminate safety monitoring for low-risk populations. So definitely it's A, as it applies to all trials, not only drug verification trial, the purpose or the role of regulatory compliance, as to confirm that the any trial meets ethical and legal standards for outcome consistency. So A would be the answer, as regulatory compliance is critical for ensuring that both validation and verification stages adhere to ethical and legal requirements, with documentation demonstrating consistent and reliable outcomes. And here we come to end of this lecture. Thank you very much and goodbye.